Am I the only one here? Oh,
You're not the only one here now. Morning.
Morning, Professor.
Yeah, I apologize. What I do is I have to click on my Zoom link and click on my Zoom link like multiple times. And I have to restart the meeting and restart the meeting until I see you. And I'm realizing it doesn't matter if I start right at 9:30 or if I start at 9:28 or if I start at 9:31. I still end up doing the same thing. And I'm not sure what the problem is. And when I've called IT Tech about it, they don't know how to tell me how to fix it either. So I apologize that we tend to start either right on time or a few minutes late. I don't know what the issue is. Nevertheless, do you have any questions for me?
Yeah, I emailed you about that one assignment that won't open up. You said it'll open up today.
Yes, it should open up. It should have opened up at midnight this morning.
Okay, thank you.
Yeah, I'm trying to make them so that and I haven't done it really well in the past, and I'm trying to get better at it. So that they're open on the same day of the lab that they're scheduled. That's the reason I scheduled it, so it wouldn't open early. And I kept thinking about your email last night, and I worked up until the wire last night. I went to bed around 1. And I was thinking about your email and thinking about trying to open it up early. And I just never got to it, and I apologize.
No, that's okay. It did open up and I just checked it.
Okay.
I have a question about the case study.
Okay.
So is basically, can we just print it out and answer the questions on the paper, draw the diagrams, and then Dropbox it, correct?
Yep.
Okay,
Yeah.
I just wanted to make sure I understood it right.
Yeah, put it as when you submit it, make sure that you submit it as either a Word document, a PDF, or a JPEG.
Okay.
People who submit Google Docs, I can't open them.
Okay.
And at least one student every class will submit things as a Google Doc.
Okay.
And I can't open them, and I'm not sure exactly why. I don't know even how to open them. When I'm asked, when I try to open them, I have to pay for software to open them.
Oh, okay.
And right now, I'm not willing to do that.
Where is our case study? Actually, I haven't.
It's actually, I'll show you where it is, but it's under case studies.
Under case studies. Okay. Is that under the content?
Yep. Go down. I think it's right underneath exams. Do you see it?
Let me look.
I'm going to click on it here.
There it is. I see it.
Other questions? Okay. I want to get through. So I went through, and this PowerPoint, we're a little behind in lecture. We're not really behind in comparison to where the labs are. In labs, we're not behind at all. In lecture, we're a little behind, but we're catching up. I think chapter 2 is just a long chapter. And it's just a very involved chapter. However, the rest of the chapters get shorter as you go. And so today's labs, they cover lab exercises 16, 17, and 19. And the first one's over the nervous tissue. Second one's over the spinal cord. And the third one is specifically looking at reflexes. Whether you recognize it or not, you're always having reflexes. Let me, Miranda probably gave the best example of a reflex, except her reflex didn't work very well. Grabbing the wrong end of the curling iron. Very few of us have to think before we let go of it. I honestly, I've never had to think before I let go of it.
I think mine was just because I grabbed it with my left hand and not my right.
But you know what? Normally you'd still let go of it. You know that? You'd still let go of it. It's a protective instinct of your body. If you think about it, it's the ability of your body to maintain homeostasis. I do know, like, I can say little tiny kids don't localize pain well enough to let go of things. I know my little niece, I remember she's now not so little. She's now as old as some of you are. She is about 22. But when she was about 2 years old, maybe a year and a half, we were out to eat and she grabbed a hot French fry. And she sat there, and I remember watching her, and she sat there and held it in her one hand and cried. Because it was, she knew she was hurting. But she, and my sister, her mother kept saying, Michelle, let it go. Michelle, let it go. Drop it. Drop it. And she wouldn't drop it. And finally her mom grabbed it from her hand and dropped it. And then she stopped crying. And little kids, because of their lack of myelination, that goes on, remember, it goes all the way, continues up through adolescence. You still, every time that you change a synapse, will undergo some form of myelination of your axons. Even after you're beyond the age of 18 or some 20. Learning is a matter of synapses reorganizing themselves. And the more you repeat, because we learn better with repetition, the more you repeat things and you hear things over and over again, that synapse becomes stronger. And you get more synapses being made on a specific connection. That's also involved in the myelination process. There's also a lot of myelination occurring. But we're going to go through this PowerPoint. Some of this you've seen before. Some of it's new because we haven't finished talking about the spinal cord in lecture. But lab exercises 16, 17, and 19. Sorry, bear with me. I am having kind of a rough morning, so I'm trying to wake myself up and get going. The central nervous system and the peripheral nervous system. The central nervous system is composed of the brain and the spinal cord, and it contains the relay neurons. The relay neurons are also called the interneurons. They're also called the association neurons. They're called the association neurons because they're connecting the sensory neurons to the motor neurons. So you sense something and you grab the wrong end of a curling iron. You sense that it's burning you. And your motor neurons fire to allow you to drop it. In that case, there's actually not an interneuron involved. But so the interneurons are normally the connections between the sensory neurons and the motor neurons. We'll talk about why there's not an interneuron in that process of grabbing the wrong end of a curling iron. What's another type? It's an autonomic reflex, but you stand up too fast. You get out of bed and you stand up too fast. We've all done this at some point. Stand up too fast. It's a low blood pressure issue, and we'll talk about it when we talk about the autonomic nervous system in lecture. But you stand up too fast. The autonomic nervous system recognizes that the pressure is lower in your upper part of your body, in your head, in your upper trunk area. Blood vessels constrict in the lower part of your body to adjust the blood pressure so it's equal. That takes a second for that to happen. That's where there's interneurons involved. So that's the difference between a monosynaptic and a polysynaptic reflex. And I'm sorry, I'm going a little bit out of order here. But a Mom, can you move? Oh, shoot. Sorry, my cat's just throwing up on a shelf. She's probably already done it. She threw up on the shelf and over here. Okay. I'm sorry, I'm just listening to my cat vomit and I knew where she was, but I couldn't get to her. So monosynaptic is when the sensory neuron is synapsing on the motor neuron and there's no interneuron involved. That's what the virtual lab is over this week. Polysynaptic are when the sensory neuron is synapsing in the spinal cord on an interneuron, and that interneuron is then synapsing onto the motor neuron. If that isn't clear, we'll go through it again here in a few minutes. So the central nervous system is composed of the brain and the spinal cord and the interneurons that are in the brain and the spinal cord. The peripheral nervous system is over the cranial nerves, which we'll talk about in chapter 14. Spinal nerves, which we'll talk about today and when we finish the spinal cord in lecture, and the peripheral nervous system. The peripheral nerves, spinal nerves are generally they're the same thing. Spinal nerves are coming directly out of the spine. When there's synapses out in the when there's synapses out in the body, in the rest of the body, besides right next to the spinal cord, those are the peripheral nerves that are being synapsed onto. Remember what a synapse is. A synapse is like, I like to think of it as a snap on a shirt. One cell's coming in conjunction with another cell. So the peripheral nervous system includes both the sensory neurons and the motor neurons. Do you have any questions for me? That was a little rough of a description. So sensory neurons, remember, these are the afferent neurons. So these are the incoming neurons. They're sensing what's occurring in the environment, what's occurring within the internal organs, and relaying that information to the central nervous system. Within the central nervous system, we have interneurons. This is also where there may be a sensory neuron that directly synapses onto a motor neuron or an efferent neuron. Remember, afferent meaning incoming information or approaching information. Efferent meaning information that's exiting out or outgoing information. So sensory neurons are specialized to detect stimuli such as heat, light, pressure, chemicals, and to transmit information about these things to the central nervous system. The sensory neurons begin in almost every organ and at the central nervous system. Interneurons, remember, these are also called association neurons or sometimes relayed neurons, make up 90% of the neurons. They lie entirely within the central nervous system. And they receive signals from many other neurons, like sensory neurons, and carry out the integrative functions of the central nervous system. Motor efferent neurons are going to send signals out to effectors such as muscles or glands. It can be skeletal muscle, it can be smooth muscle, or it can be glandular tissue. So three basic types of neurons. Sensory neurons, which receive information from the environment and the internal organs. Synapse with an interneuron within the central nervous system, which then will synapse with a motor neuron that will synapse with effectors. We'll talk about this again in a second, but this is a typical reflex arc. One second here. This is a typical reflex arc. You've seen this before when we talked about feedback loops, because this is a feedback loop. Reflex arc. Has 5 components. Receptor, sensory, afferent neuron, as I can't type. interneuron or an integration center. Remember, we also call these an association or relay neuron. Or a motor, or an effector neuron. And an effector. Oh, I said effector, efferent. Effector. So these are the 5 components of a reflex arc, which is the same thing as any time that the peripheral sensory nervous system is relating with the peripheral nervous system. So you can say that many things in your body follow this specific pattern. Reflexes are a little different in that it's always an unconscious effort. Sometimes when we use this pattern, it's a voluntary thing. You think about it first. In reflexes, this is involuntary. It's all happening under unconscious control. You have questions for me so far? So a general review of the structure of a neuron. The neuron body starts with the cell body. The neuron starts with, and remember, this is also called the soma, which includes the nuclei, the mitochondria, and the rough endoplasmic reticulum. Dendrites that receive messages from other cells. Axons, which pass messages away from the cell body to other neurons. Sometimes axons are myelinated and sometimes they're not. Remember that it's saltatory conduction along a myelinated axon and continuous conduction along an unmyelinated axon. And the terminal branches of the axon, this is where a neuron will form junctions or synapses with other cells. Junctions in this case is synonymous with synapses. Dendrites are the extensions of the cell body or the soma. I like to think of these as being little feelers. They They're very flexible. You can develop more dendrites at a neuron the more often that neuron is stimulated. I think I've told you before, and I don't wear it during this class because the noises around me can distract me. But with my face-to-face classes, I wear a hearing aid because it just makes it easier for me to hear my students because I'm hearing impaired. And I grew up being hearing impaired. I was diagnosed at about 18 months as being hearing impaired. And I didn't start wearing a hearing aid until I was about right around 30 years old, somewhere right in that age. And when I talked with my audiologist the first time, he made the comment to me. He said, when you start wearing a hearing aid, he says, you're going to recognize and you're going to learn over time that your hearing will get better even when you're not wearing a hearing aid. And one thing that, I learned really quickly when I got my first hearing aid is, I was hearing sounds that I'd never heard before. I was hearing, I remember walking out of the office and hearing the music in the lobby that I know I hadn't heard before. And being able to hear the clicker in my car when you signal to make a turn. And little things like that. And now I realize that I hear better even when I'm not wearing my hearing aid than I did before I ever got the hearing aid. The reason for that is because the hearing aid works as a little microphone. Mine stays in my ear, and it is just a little microphone that just amplifies the noise around you and sends it through to your tympanic membrane, and it just amplifies it so that the nerves in your ear are picking it up better. When a nerve is stimulated more, when the nerve is meaning the nerves in there are picking it up better, they're being stimulated more, they develop more dendrites. However, I did go through an experience recently where I gave up my hearing aid got broken, and it was kind of a bad experience for me. And hearing aids are not cheap. My little hearing aid is it's about the size of a dime. It's maybe a little smaller than a dime. And next to my car, it's the most expensive thing I own, because this little one that I have right now cost me $3,500. And so the reason I say all that is because I had one that it got broken after 4 years. And it got sent back to the manufacturer and got sent back to me broken. And when I took it into an audiologist, they told me that they just break after 4 years, which really annoyed me because it wasn't that broken before I sent it in. But so I quit wearing the hearing aid for about a year and a half. And just this last summer, I got another one. And I'm realizing that I'm going through it all over again, where I hear better even when I'm not wearing it. I could tell that my hearing was going down when I wasn't wearing my hearing aid for a year. And the reason I'm bringing all this up is to let you know how flexible neurons are with regards to these dendrites and the myelination. the more you stimulate them, the more developed they become. If you stop stimulating them, they tend to atrophy a little bit. And then you can, a lot of times you start stimulating them again and they become more developed again. But so with this neuron body, here's our soma, here's our dendrites, here's the neuroglia, here's the axon that's going to come out and terminate in the terminal arborization or the axon terminal. Myelinated axons have this spiral layer of insulation around the axon that's formed by the oligodendrocytes in the central nervous system and the Schwann cells in the peripheral nervous system. The myelin is like that of a plasma membrane. So remember that it's got the same consistency of the plasma membrane. So here's our axon. Here's the myelin sheath. The most outer layer of the myelin sheath is the neural lemma. Unmyelinated axons are enveloped in the neural lemma of myelinated axons. So here's our axon. Here's the myelin of that axon. The most outer layer is the neural lemma, in which the unmyelinated axons are enveloped in. So look in here. Here's the myelination of an axon. Here's the axon. Here's the myelin. In the most outer layer are unmyelinated axons. You're seeing them right here. Here's an axon that's unmyelinated. Here's another one. Here's another one. We know that the presence or absence of myelin allows for faster conduction. So remember, it's a presence or absence of myelin and the diameter of the axon. The larger the axon, the faster the conduction. Whether there's myelin or not, the more myelin there, if it's myelinated, the faster the conduction. A neuron dendrites are the input zone, or it's also the input zone is also the soma, is where neurons receive information, incoming signals. The trigger zone that is about the same place as the axon hillock is going to be where we're going to either initiate where we can initiate action potentials. The conducting zone, the myelinated or unmyelinated axon. And the axon terminals. You have questions for me? when we talk about signal conduction, there's 3 kinds of channels that open and close in response to stimuli. We have chemical channels that respond when a ligand binds to them. And that ligand can be neurotransmitter, solute molecule, it can be a lot of different things. We have mechanically gated channels which move the open in response to pressure. Or they can get bumped, they can get pulled open. It's a matter of a physical push. And then we have voltage gated channels that open at a certain voltage and they close at a certain voltage. The resting membrane potential in a neuron is typically about minus 70 millivolts. So the inside of this neuron is approximately minus 70 millivolts more negative than the outside of the neuron. If you think about this, this is a potential energy source that can be used to do work later, like conducting a signal. That's why we call it a membrane potential. What factors are involved in maintaining the resting membrane potential? Well, potassium ions have the greatest influence on the resting membrane potential, because the plasma membrane is most permeable to potassium than it is to any other ion. Potassium diffuses freely through ion channels, leaving larger negatively charged anions behind. So proteins, negatively charged proteins, are big. They're not going to move through the membrane. So they get trapped in the membrane, inside the cell. So they generally are what's keeping the inside of the cell more negative. All the time. They're not moving. However, the fact that the potassium can move back and forth, some of it will be attracted and stay with the anions, the negatively charged anions inside the cell. Another 2 factors that are involved in maintaining the resting membrane potential is that sodium is 12 times more concentrated in the extracellular fluid than it is in the intracellular fluid. However, sodium tends to leak very, very slowly back into the cell because there are leak channels. But it's still overall, because of the sodium potassium pump, overall it's still 12 times more concentrated outside of the cell than it is inside of the cell. So the major factor that is important in maintaining the resting membrane potential is the sodium concentration. Sorry, not the sodium, the potassium concentration. One thing that keeps these things constantly, with sodium always being high, more concentrated outside of the cell, more potassium being concentrated inside of the cell, is the sodium potassium pump. Questions? So neurons using this resting membrane potential communicate via two different types of electrical signals: the graded potentials and the action potentials. Graded potentials, remember, they're the ones that die off with distance, and they can be hyperpolarizing or depolarizing. Action potentials are always depolarizing, and they communicate over long distances where they don't vary out in amplitude. They don't get weaker as they move farther away from the origin. The amplitude remains the same. So an action potential is a rapid up-down shift in the membrane's potential that is produced by the opening of voltage-gated channels in the plasma membrane. Questions? So the beginning of an action potential is where, whether or not, let me start differently here, whether or not an action potential will occur is a matter of whether or not the threshold membrane potential is reached. This involves the summation of inhibitory or hyperpolarizing postsynaptic potentials and excitatory postsynaptic potentials. If the threshold is reached at about minus 55 millivolts, sodium channels will open and potassium channels will begin to open. But the sodium channels open a lot faster. As the sodium rushes into the cell, more sodium channels open up. And as more sodium channels open up, that's when we get this very fast, sharp depolarization that will continue until the inside of the cell is actually a positive 35 millivolts or positive 30 approximately. So now the inside of the cell is positive instead of negative. At this point, the sodium channels are going to close because this is the voltage at which they close. They'll start to close. And the potassium channels that started to open at the beginning are now fully open. And so potassium starts to leave the cell, making the cell more negative. And it will continue to leave the cell until they start to close at the minus 70 millivolts. They're slower to open and they're slower to close, potassium channels, which means that we get a hyperpolarization of the membrane. Now I want, actually let me go through this first and then I want to show you a short video here. Comparison of local potentials. Remember, these are the graded potentials versus action potentials. Local potentials are produced by gated channels on the dendrites on the soma. Action potentials are produced by voltage gated channels on the trigger zone and axon. Local potentials may be positive or negative. It can be depolarizing or they can be hyperpolarizing. Action potentials always begin with being depolarizing. Local potentials are graded, meaning they're proportional to the stimulus strength. Action potentials are all or none. Either it is going to occur and it's going to occur to the maximum amplitude, or it's not going to occur at all. Regardless of what the initiating stimulus was. Local potentials are reversible, meaning they will return to the resting membrane potential as soon as the stimulus ceases, even before threshold is reached. Action potentials are irreversible. Once they start, they go to completion. Local potentials are local, meaning they're only active for a very short distance from where they started. Action potentials are self-propagating. They are involved in great distances from the point of, they move over great distances from the point of origin. Local potentials are decremental, meaning the signal grows weaker with distance. Action potentials are non-decremental, meaning the signal is going to maintain the same strength regardless of the distance. Oh, I want to pause right there for a minute. I have this short video clip talking about resting membrane potentials and action potentials.
This is the checkers game where grandson and granddad will bond.
Neurons have three different kinds of potentials: resting, graded, and the action potential. First, let's see what happens at resting potential, which is at -70 millivolts. The neuron maintains this potential due to differences in permeability of ions on either side of its cell membrane, as well as the sodium-potassium pump. The ions contributing to the charges on either side of the membrane are negatively charged proteins, negatively charged chloride, positively charged sodium, and positively charged potassium. Now, there are several kinds of channels found in cell membranes, allowing for the transport of substances from one side to the other. Two kinds are important in the context of action potentials: leaky channels and voltage-gated channels. Leaky channels allow the free flow of substances through them. Voltage-gated channels are only open at certain voltages. Back to the ions, chloride and the proteins stay put, as they don't have a means to cross the membrane. However, the neuron does have leaky sodium and potassium channels. These are always open and allow some flux of these ions. So what do the ions do? First, there's a lot more potassium in the cell than outside the cell. The potassium wants to rush out of the cell because of the chemical gradient, but wants to stay in the cell because of the electrical gradient. Like charges repel. Similarly, there's a lot more sodium outside the cell than in it, and the sodium has its own electrochemical gradient. Now, if you were to just let the ions do their thing and flow through these leaky channels, eventually a bunch of sodium would rush in, and we'd lose that potential. However, the sodium-potassium pump is at work. The energy from one ATP hydrolysis reaction allows this pump to transfer 3 sodium ions out and 2 potassiums in. As a result, the neuron maintains its resting potential of -70 millivolts. A graded potential is a change in potential that can vary in size, with magnitude depending on the intensity of the stimulus, and occurs when the neurons get excitatory postsynaptic potentials, or EPSPs, or inhibitory postsynaptic potentials, or IPSPs. Dendrites are a neuron's input zone, where a neuron can get EPSPs and IPSPs from other neurons. The neuron cell body is like a calculator, integrating these signals. When the summation of graded potentials results in a potential of -55 millivolts at the axon hillock, an action potential occurs. Hence, EPSPs make it more likely that an action potential will occur, while IPSPs make it less likely. Unlike graded potentials, which are changes in potential varying in size, action potentials are all or nothing. More intense stimuli simply mean a higher frequency of firing. So let's go through the steps of an action potential. We start off at resting potential. Voltage-gated sodium and potassium channels are closed. Then, a stimulus causes some voltage-gated sodium channels to open. Once we get to the threshold of -55 millivolts, the action potential begins, with lots of other voltage-gated sodium channels opening. With sodium channels open, depolarization occurs. Sodium rapidly rushes into the cell, and the voltage soars up to +30 millivolts, at which point the voltage-gated sodium channels close. Potassium channels now open, and repolarization occurs as potassium rapidly rushes out of the cell. The voltage zooms down and overshoots the -70 millivolts before the potassium channels can close. Finally, the sodium-potassium pump restores the resting membrane potential. It does a conformational change thanks to an ATP molecule being hydrolyzed. Again, this conformational change results in 3 sodium atoms being shuttled out of the cell and 2 potassium atoms being shuttled in. We've now seen what happens locally at one segment of the axon, but how does the action potential propagate? Well, when the sodium ions are rushing in during depolarization, they repel each other and so spread out. This makes the next section of the axon reach threshold and also have an action potential. Why doesn't the action potential travel backwards, though? At the same time as the first section is depolarizing, the section of axon behind it is experiencing repolarization, and the potassium rushing out results in a refractory period. There is an absolute and relative refractory period. During the absolute refractory periods, which coincides with most of the action potential's duration, you can't trigger another action potential, because the sodium channels are briefly inactivated. The membrane needs to be hyperpolarized before that can happen. During the relative refractory period, only a very strong stimulus can cause an action potential, so since the membrane is below the resting potential and you need a stronger EPSP to get to threshold. Another important point, action potential propagation is slow. That's why most of your neurons have myelin sheaths, which are rich in lipids. Myelin sheaths are made by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. They insulate the axon and prevent leakage of charged ions. Instead, you get what's called saltatory conduction, in which action potentials only occur in the spaces between the myelin, called nodes of Ranvier. In addition to myelin sheaths, neurons with larger diameters also have faster transmission speeds. Once the action potential reaches the end of the axon, there are terminal buttons there. The depolarization triggers the opening of voltage-gated calcium channels on the presynaptic membrane.
Actually, we'll just finish this out because this is what we were going to talk about next.
And calcium rushes into the terminal button. This causes exocytosis of vesicles full of neurotransmitter molecules, and neurotransmitter is released into the synaptic cleft, where the neurotransmitters attach to the receptors of the postsynaptic neurons, causing EPSPs or IPSPs, and potentially triggering another action potential.
If you like this video, please like and subscribe. It would help me make more videos. And
Questions for me? Let's do something right now. Let's take a 10-minute break right here. And we'll move forward from here. So we'll come back at 10:43. Okay, so let's start here. So synapses, we have two different types of synapses. We have chemical synapses, where neurons are going to communicate via neurotransmitters, or we have chemical electrical synapses, where adjacent cells are going to be connected to one another by gap junctions. made of connexins. We talked about this other day, but connexins are essentially half of the tunnel on each part of the membrane. That come together to make a complete tunnel from one cell into the next cell. So signals arrive at the presynaptic neuron. They arrive at the terminal of the presynaptic neuron. The postsynaptic neuron is going to respond to the signal. And presynaptic neurons can synapse with different parts of the postsynaptic cell. They can synapse with a dendrite, a soma, or the axon. We will spend most of the time when we talk about the nervous system, we're referring to chemical synapses. Electrical synapses, like I said, they tend to, like I said the other day, they tend to be between cardiac muscle cells, between smooth muscle cells, allowing things to all contract at the same time and relax at the same time. When you talk about the heart in 122, in biology 122, in the next semester of A&P, you will learn about that the heart has a lot of its own electrical wiring system that's involved in those synapses. So those are electrical synapses. So the majority of what we talk about with regards to the nervous system this semester is referring to chemical synapses, meaning where messages are being transferred from one neuron to the next neuron via neurotransmitters. So neurotransmitters are molecules that are synthesized by a neuron and released when a nerve signal reaches an axon terminal. And they have specific effects on the target cell's physiology. Different neurotransmitters can have different effects based on the receptor on the postsynaptic neuron. We can have excitatory postsynaptic potentials, which is a voltage change that makes a neuron more likely to initiate an action potential because it's a depolarizing potential. So sodium is going to flow into the cell, making the inside of the cell less negative, therefore making it more depolarizing it. Or it can be an inhibitory postsynaptic potential where it's hyperpolarizing the memory, making it more negative than it was before. Inhibitory postsynaptic potentials occur because chloride is flowing into the cell, which is negatively charged, making the inside of the cell more negative, or potassium, the positively charged ion, is leaving the cell. These different excitatory postsynaptic potentials and inhibitory postsynaptic potentials can summate, which means that they can add together to allow for the membrane potential to reach threshold, or they can neutralize one another. And a depolarizing potential and a hyperpolarizing potential can cancel each other out. So when potassium leaves the cell, it's going to result in an inhibitory postsynaptic potential because it's making the inside of the cell more negative and pushing it for the membrane potential further away from threshold. When chloride enters a cell, because it's negatively charged, it's going to make the inside of the cell more negative. Causing the cell to become more negative than it was before. Moving further away from reaching the possibility of reaching threshold. When sodium enters a cell, it's going to result in an excitatory, meaning depolarizing, allowing the cell to be more inclined to reach threshold with regards to the membrane potential. So remember that these can add up, and over time we can possibly reach threshold and fire an action potential. So altogether, these don't automatically say that an action potential is going to be fired. They just increase or decrease the likelihood that an action potential will occur. Questions? Guys are really quiet this morning. So moving on, talking about specific organs of the nervous system. The spinal cord is a cylinder of nervous tissue that arises from the brainstem at the foramen magnum of the skull. It occupies about 2/3 of the vertebral canal, ending at about L1, L2. Because the vertebral column keeps growing and the spinal cord does not. That's the reason the vertebral column keeps growing and the spinal cord does not. That is why the vertebral column is longer than the spinal cord. The spinal cord is going to give rise to about 31 pairs of spinal nerves. Each part of the cord is going to be supplied. So each part of the body is going to be supplied by each pair of spinal nerves is called a segment. So cranial nerve number 1, there's 2 of them, and that's 1 segment. Cranial nerve number 2 is 1 segment, and there's two of them, one coming out each side through that intervertebral foramen. The L3, cranial nerve number L3, there's 2 of them, they come out each side. In the spinal cord, there's 2 major enlargements. There's a cervical enlargement which gives rise to the nerves that are going to innervate the upper limb, your arms. And a lumbosacral enlargement, which is going to give rise to the nerves of the pelvic region and the lower limbs. The conus medullaris is what it sounds like. It's a cone. It's the terminal end of the spinal cord. The tip of it gives rise to the cauda equina, which if you're a horse person like my mom who's on the phone in the other room, it's equine means a horse, cauda meaning tail. So this kind of looks like a horse's tail. Gives rise to a bundle of nerve roots that occupy the canal from L2, the L2 vertebrae, to the S5, the fifth sacral vertebrae. I wanted to add some pictures because I learned anatomy using cadavers. And I think sometimes our pictures that we have and the diagrams we have don't do anatomy a lot of justice. It's a lot more involved than what we see on a diagram. So this is the tip of the spinal cord. So we're starting right about, let's see here. I would say this looks like it's about starting at about T11. Right here is the conus medullaris. This sheath is the dura mater. And on the tip, starting at the tip and kind of a little further, more anterior, assuming more superior to the conus medullaris, is the cauda equina. Little meningeal sleeves. What do you think meningeal sleeves are? What are the meninges? Someone tell me, what are the meninges from the other day? Jamie, what are the meninges?
Isn't that what? Oh,
And what are the meninges?
isn't that what protects the spine?
It does. So they're just sheets of dense, irregular connective tissue. And there's 3 of them, and they're protecting the spine. If you look here, they're also involved in the terminal or the initial parts of the spinal nerves that are coming out. If you think here, these terminal sleeves would be the spinal nerves coming out through the intervertebral foramen of the spinal cord, of the vertebral column. So they're just little extensions of the meninges that cover the first part of the spinal cord as it's exiting the spinal cord. So the spinal nerves are divided into 4 regions. The cervical spinal nerves, the thoracic spinal nerves, the lumbar nerves, spinal nerves, and the sacral spinal nerves. There's 8 pairs of cervical nerves. So they control the sensory and motor activity of the head, the neck, the upper body, the arms, and the hands. The thoracic nerves are controlling the sensory and motor activity of the hands, the fingers, the chest, and the abdominal muscles. The lumbar nerves, 5 pairs, control the sensory and motor activity of the hips, the knees, the ankles, and the toe muscles. The sacral nerves are going to be the sensory and motor activity of the legs, the toes, the bladder, and the anal muscles. And then there's 1 pair of coccygeal nerves. The meninges is the dura mater, the arachnoid mater, and the pia mater. Remember that in the meninges that surround the spinal cord, we have an epidural space. We'll learn when we talk about the meninges of the brain that we don't have an epidural space there. We only have it in the spinal cord. The arachnoid mater looks like a spider webby type of material that adheres to the dura and is separated from the pia by fibers spanning the subarachnoid space, which is filled with cerebrospinal fluid. That subarachnoid space is where they would do a spinal tap to detect for meningitis. The pia mater is a delicate membrane that's composed of 1 or 2 layers of squamous to cuboidal cells that's collagenous and elastic fibers that supplies blood vessels that supply nutrients and eliminate waste. If you remember, I told you that the pia mater is very, very thin. If you think 2 layers of cells, it's going to be very, very thin. So you're essentially looking through it when you looked at this, you're looking through it at the spinal cord. Here's the dura mater. Denticulate ligaments are little thickenings of the pia mater that suspend the spinal cord and the dura mater sheath. So little attachments between the pia mater and the dura mater. Do you have questions for me so far? The epidural space is like epi always means on top. So this is the space above the dura mater. Here's the dura mater, here's the epidural space. Here's the arachnoid mater. This second layer right here. Here's the subarachnoid space. And then the pia mater is right on top of the spinal cord. looking at a cross section through the spinal cord, the central area is gray matter. This area right through here is all gray matter. That's surrounded by white matter that is arranged in 3 funiculi. I'm going to stop sharing just for a second here. Funiculi, and I think your the manual actually refers to them as being white columns because they are white matter. So funiculi are columns of white matter. What is the white matter? What do you remember what we called white and gray matter? What was the white matter? What was the white matter?
Myelin?
It was myelin. So it's myelinated axons. Jessica, you're right. It's myelinated axons. We also, when we're referring to axons or bundles of axons in the central nervous system, we're calling them tracts. So we have this posterior funiculi, meaning a white column of tracts in the posterior surface of the spinal cord, a lateral funiculi, and an anterior funiculi. One thing that we also see here is we have an anterior median fissure. So anterior meaning in the front, median meaning in the middle, a fissure meaning an indentation. In the back, it's not labeled here, but this is the posterior median fissure. So gray matter is neuron cell bodies with very little myelin. White matter is abundantly myelinated axons. When we look specifically at the gray matter, we're looking at pairs of posterior or dorsal horns. Here's the dorsal horns. Remember, this is the posterior surface. So these are the posterior horns, which contain the posterior root of the spinal nerve with the sensory fibers. Keep this in mind for a second. So posterior horn is going to contain the spinal roots of the sensory fibers. The anterior or ventral horns carry the motor fibers. Remember we talked about that, the relationship between sensory neurons, interneurons, and motor neurons. So sensory fibers are coming in through the posterior horn, interneurons in here. Motor neurons are coming out of the ventral horn. This is key. The gray commissure is the link between the two halves of the spinal cord. Within it, and it's not labeled here, actually, nope, it's not labeled here. There's a central canal, which is filled with, lined with ependymal cells. Remember, those are the neuroglia that secrete cerebrospinal fluid. And so this central canal is also full of cerebrospinal fluid. The lateral horn that's only visible from T2, the thoracic vertebrae 2, to L1, the lumbar vertebrae 1, contains the cell bodies of the motor neurons involved in the sympathetic autonomic nervous system. So we only see it from T2 to L1. It's going to be important when we talk about the autonomic nervous system. That the lateral horn is only involved in the sympathetic nervous system, autonomic, that's part of the autonomic nervous system. So the white matter of the spinal cord includes bundles of axons or tracts that go up and down the cord, allowing for communication between different levels of the central nervous system. Remember, here we're just looking through a cross section. There's these bundles of white matter, or bundles of axons, tracts, are arranged in 3 pair of funiculi. The posterior or dorsal funiculi, the lateral funiculi, and the anterior funiculi. We have ascending tracts in these funiculi, and we have descending tracts. The ascending tracts are going to conduct sensory nerve impulses up the spinal cord toward the brain. So these are specifically sensory, so bringing in information into the central nervous system about the environment and the internal organs. Descending tracts. So the ascending tracts are the red, descending tracts are the green. Descending tracts are going to conduct motor nerve impulses down the spinal cord from the brain. So these are our motor saying this, these are the, if you think about the reflex arc, ascending tracts are going to bring in the input from the sensory neurons. Synapse in the integration center. Descending tracts are going to be the motor nerve impulses that are going to lead out to the effectors. You have questions for me? So each of these tracts, each of these bundles of axons are going to carry specific types of information from one location to another. If you want to carry painful information from the spinal cord to the brain, you're going to use one particular tract, the spinothalamic tract. So one bundle of axons, which are going to be ascending because it's going to be painful information about the environment or about your visceral organs. So it's sensory information. If you want to carry voluntary movement commands from the brain to the spinal cord, you're going to use a different tract. So this is going to be motor because voluntary movement is motor, which is going to be the lateral corticospinal tract. Some of these tracts can undergo decussation. What this means is that they can cross the midline, so that the brain senses and controls on the contralateral or the opposite side of the body. So some motor commands of your body are initiated by the right side of your so to me some of your left motor commands, left motor movements are controlled by the right side of your brain. Some of the motor movements on the right side of your body are controlled by the left side of the brain. Decussation. If tracts do not decussate, they're ipsilateral, meaning they're on the same side. Do you have questions for me so far? So ascending tracts carry sensory signals up from the spinal cord. Sensory signals can travel across 3 neurons from the origin, the receptors, to the destinations in the different sensory areas of the brain. The first order neurons detect stimulus and transmit a signal to the spinal cord or brainstem. So first order, coming from the stimulus and transmit it to the spinal cord or the brainstem. Second order neurons are going to continue to the thalamus at the upper end of the brainstem. We'll talk when we talk about in chapter 14, which we will start next week. We'll talk about the thalamus and then it's part of the internal part of the brain. But second order neurons are going to continue from where the first order neurons drop off in the spinal cord or the brainstem. And our second order neurons are going to continue to the thalamus. Third order neurons are going to carry the signals the rest of the way to the sensory region of the cerebral cortex. Cerebral cortex meaning when we talk about a cortex, we're referring to the outer edge or the outer covering. So in the case of the cerebrum, the cerebral cortex is the outer edge. Let me ask you something really quick, and I know this is kind of going off on a tangent, but if this is the brain, this is a cross section through the brain, where's the white matter, where's the gray matter? White matter looks white, gray matter is not going to look so white.
Gray matter looks red.
Or it looks kind of tan.
Yeah.
So how does this compare to the spinal cord?
Would it be the same?
You know, go back a couple slides. Look at the gray matter. Where's the gray matter in the brain? In the
Oh.
spinal cord. I'm sorry, where's the gray matter in the spinal cord?
In the center.
It's in the center. So it's the reverse. So in the spinal cord, the cell bodies are in the internal spinal cord. And the ascending tracts are on the outside. In the brain, the white matter or the axon, myelinated axons connections are in the inside and the cell bodies are on the outer surface. So the ascending tract, remember, we can go through first order, second order, and third order neurons, lies within the anterior and lateral funiculi. Remember, these are the white columns of myelinated bundles of axons or tracts. The spinal thalamic tract transmits sensation of pain, warmth, coolness, tingling and itching, and deep pressure. So this is all sensory. If you think pain, warmth, coolness, tickling, tickling is actually a feeling of pain. Itching and deep pressure. This spinal thalamic tract is made of second order nerve fibers that travel up the spinal cord to the thalamus. Due to the crossing of the second order neurons, these signals are sent to the contralateral, crosses over down here, crosses over to the contralateral side of the cerebral hemisphere. So this is going to decussate. In contrast, descending tracts are going to conduct motor signals down the brainstem and spinal cord. Upper motor neurons are going to originate in the cerebral cortex or brainstem and terminate on a lower motor neuron. The lower motor neuron can be in the brainstem or the spinal cord. The axon of the lower motor neuron is going to lead to muscles or other target organs. So we have, rather than having 3 different orders of neurons like we have in ascending sensory tracts like the spinal thalamic tract, in the descending tract we have an upper motor neuron and a lower motor neuron. Remember, they call this is also the corticospinal tract. This is one of the descending tracts of the corticospinal tract. Excuse me, one of the descending tracts, that's one of the motor neuron tracts, is the corticospinal tract. So it's going to conduct nerve impulses from the cerebral cortex to the spinal cord for voluntary movement of skeletal muscles. it's going to conduct nerve impulse. Excuse me, I've already said that. If your brain decides that it wants your legs to move, it's going to send this voluntary movement command from the brain down to the region of the spinal cord that's responsible for leg movements. I'm getting my teaching assistant back. Questions? It's talking specifically about the nerves. The spinal cord communicates with the rest of the body by way of these spinal nerves. A nerve is a cord-like organ that's composed of nerve fibers bound together by connective tissue. Bundles of axons within the spinal nerve are fascicles that are bound by a perineurium. These different layers, I'm going to stop sharing just for a second here. Neurium. Neurium. Yeah, neurium. It's going to be layers or connective tissue layers that surround the spinal nerve tracts. It's always going to refer to layers around this nervous system, around the nerve tracts. Tracts or axons. So epineurium is going to be the outer layer, epi meaning on top. So this is going to be the outer connective tissue. Perineurium is going to be what's going to be the sheath or the connective tissue membrane around one bundle of axons. Around each particular myelinated axon, there's an endoneurium. So endoneurium around one individual myelinated axon. Perineurium around bundles of fascicles or bundles of axons that make up one fascicle. And epineurium. So sensory afferent nerves are going to carry signals from the sensory receptors to the central nervous system. Motor or efferent nerves are going to carry signals from the central nervous system to muscles, cells, and glands. Mixed nerves are bundles of sensory and motor nerve fibers. So if I'm looking at one of these nerves that's coming from the spinal cord or entering the spinal cord. One of these fascials may be involved in sensory nerves. One may be involved in motor. If this is the case, if individual fascials are involved in sensory and motor, and they're combined in one nerve, that's a mixed nerve. So this would be one spinal nerve. And within each spinal nerve, we have fascials, where one fascial may carry motor nerve fibers, and another one carries sensory nerve fibers, and another one carries motor nerve fibers. When we reach this point right here, the sensory nerve fibers are going to move in through the posterior side of the spinal cord, and the motor nerve fibers are going to come out the anterior part of the spinal cord, or the anterior horn. So sensory is going to come in the posterior side, or the dorsal root, dorsal horn. The motor nerve fibers are going to come out the anterior horn, the ventral root. A ganglion is a cluster of neurosomas. What are neurosomas? Somebody tell me, what are neurosomas?
Cell bodies.
They're cell bodies, right, Rachel? So a ganglion is a cluster of cell bodies outside of the central nervous system. The posterior root ganglion contains the sensory neuron cell bodies. Of the sensory neurons. So the dorsal root is coming in the dorsal side of the spinal cord, is a sensory input to the spinal cord. The anterior root is the motor output of the spinal cord. Right near the posterior horn is what we call the posterior root ganglion. We talked about that before, right here. It's right here, which contains the cell bodies of the sensory neurons carrying signals to the spinal cord. Six to eight rootlets from the dorsal root ganglion or the posterior root ganglion are going to converge to form the dorsal root that's going to enter via the posterior horn. anterior root is the motor output out of the spinal cord, where 6 to 8 rootlets is going to leave the spinal cord and converge to form a ventral root. So dorsal root is going to carry sensory. The anterior root or the ventral root is going to carry motor. Remember, a posterior ramus of the spinal nerve. So remember, a ramus is an arm. It's an extension. The posterior ramus of a spinal nerve. So here, it's kind of small here, but here's the dorsal root ganglion. Here's the anterior horn. It's going to become the anterior root, which is going to be the motor output. The posterior ramus is a branch of each spinal nerve that's going to innervate certain parts of the skin, muscles of the neck, and deep muscles of the back. I'm going to do deep muscles of, so this is a very back part of your back. The anterior rami this part right here is in the thoracic region is going to give rise to intercostal muscles. Intercostal meaning in between the ribs. So these are the nerves that are going to innervate the intercostal muscles. So the anterior ramus in the spinal nerve area or in the thoracic nerve area is going to innervate the intercostal nerves. And just to put this all in a little bit of context, and because it's like I said, some of these diagrams don't do this justice. Here's looking through a cadaver, a cross section through a cadaver. So here's your posterior root. Here's your anterior root. Bear with me here. Oh, I messed it up. Here's your anterior root. Here's your posterior root. Because this is your vertebrae, so it's got to be on the posterior surface. Anterior rami. Posterior rami. Epidural space. Right here, approximately right here would be the dura mater. So it would be your anterior median sulcus or fissure. So it would be your posterior median fissure. Right here would be your gray commissure. So anterior rami, posterior rami, epidural space. This area all in here, this empty space right here. Is your subarachnoid space. Guess that this right here, so here's the epidural space, so this has to be the derm model right here. Questions? Rami communicantes. So these are branches of each spinal nerve that contain axons related to the autonomic nervous system. They consist of a pair of rami, one white ramus and one gray ramus. Rami communicantes. They're going to be involved in the autonomic nervous system. Questions? So the ventral or anterior rami are going to form networks of nerves called plexuses. The intercostal or thoracic nerves are not going to enter plexuses, but directly innervate spaces they supply in the intercostal structures they supply in the intercostal spaces. Intercostal meaning in between the ribs. So that's the reason we have a cervical plexus up here. We have a brachial plexus right here, brachial meaning the arm. Lumbar down here, sacral and coccygeal, but we don't have a thoracic plexus. So plexuses are organizations of spinal nerves. The cervical plexus, this organization that C1 is made from the C1 to C5 spinal nerves, are going to innervate and be involved in the motor signals. So sensory and motor for skin, muscles of the head, neck, upper part of the shoulders, and the diaphragm. The phrenic nerve, which is a vital nerve, is going to arise from the cervical plexus. It's the major nerve that innervates the diaphragm. Why do we care about a diaphragm? The diaphragm is the muscle the major muscle that's involved in allowing you to breathe. So a cervical break above C3. So let me backtrack just for a second. So here's C1, C2, C3, C4, and C5. Here's the phrenic nerve. So C3, C4, and C5. A break above C3 or superior to C3 is going to cause loss of innervation to the diaphragm. I have to remember his name and I can't remember it. But if you ever watched, and I know you, most of you are fairly young. Bear with me just for a second. And I wish I could remember his name because that would help me out, but I can't remember it. The old Superman movies. Christopher Reeves, thank you, Mom. Christopher Reeves. So Christopher Reeves was the man who was the major actor in the originating Superman movies. Some point, and I think it'll tell me the accident. Oh. In 1995, he was left paralyzed from the neck down after being thrown from a horse and breaking his neck during an equestrian competition in Virginia. My mother says jumping, she's the horse person. So break between C3 and C2 are right underneath it. And it was kind of a big deal because this was Superman. And so 1995, and he died, I don't know how long ago he died, but it was several years ago. So just anytime there's a break, everything that's below that break is going to be, the function is going to be lost. So that's why part of the reason we care about these plexuses. So on a model, here's the cervical plexus. Here's the phrenic nerve that's going to come down and innervate the diaphragm. The brachial plexus includes 5 major nerves: the musculocutaneous, the axillary, the radial, the medial, and the ulnar nerve. These nerves are going to supply the neck, the shoulders, and the upper limbs. One of these nerves is your funny bone nerve. We talked about this before, so don't do it, but banging your ulnar, your medial epicondyle of your elbow is going to crush the ulnar nerve up against the bone and cause that tingling. Right here. So this is the ulnar nerve is coming off the brachial plexus. The median nerve is the one that people struggle with carpal tunnel syndrome. Brachial plexus. The lumbar plexus is going to supply the abdominal wall, the anterior lateral abdominal wall, the external genitalia, and part of the lower limbs. The largest nerve coming off the lumbar plexus is the femoral nerve. And if you think back, the femur, so the femoral nerve is going to run down the femur. Damage to the femoral nerve is going to result in a loss of sensation in skin over the anterior medial aspect of the thigh. So lumbar plexus, here's the femoral nerve. Another one that's kind of important here is the obturator nerve, which is, I think, this one right here. That's going to go through that obturator foramen in the pelvis, or the os coxae. The sacral nerve is going to supply to the buttocks, the peritoneum, which is the peritoneal cap membrane around the, actually, no, it's not. Peritoneum is your anal canal area and part of the lower limbs. It's going to give rise to the largest nerve in the body, the sciatic nerve. If you've ever heard of someone struggling with sciatica, this is a pinching of the sciatic nerve. So it's a numbness that is going to run down the back of your leg. Wherever these nerves run, they innervate what's there. So if a nerve runs through a particular muscle, it's going to innervate that muscle. If it moves by the skin of a certain area of the body, it's going to innervate that skin. Here's the sciatic nerve. You remember when we talked about the bone markings, the os coxae and the greater sciatic notch. The sciatic nerve runs through the underneath in that greater sciatic notch. Please let me know if you have questions. Do you have questions? Last part we're going to talk about is reflexes. And I really do think we're going to get out early today. And I will let you go. We're not going to have time to do anything with lecture. So I'll just end class. But let me talk about reflexes first. And then a lot of this, we will do a little bit of a review for next Tuesday, and then we will start the brain. Because you'll see that a lot of this we also are going to go over in lecture. A lot of lab should be a review and an application of what's occurring in lecture. So reflexes are these quick, involuntary, stereotyped reactions of glands or muscles to stimulation. Reflexes require a stimulation. They're not spontaneous actions, but they're responses to a sensory input. They're quick. They can involve few, if any, interneurons and minimum synaptic delay. Remember, there's a synaptic delay a lot of times with the synapse because the neurotransmitter has to be excreted, or exocytosis, it has to be released, then it diffuses across the cleft and binds to the receptors, and then it stimulates an action. Reflexes are minimized, have very minimal synaptic delay. Because many times there is not an interneuron involved in the central nervous system. The reflexes are often involuntary. Actually, let's say they're not often involuntary. Actually, let me say it this way. Most often they're involuntary. They're difficult to suppress. Can you suppress a reflex? Yes, you can. But they're difficult to suppress at the same time. No. Sorry, my one little kitty is chewing on my computer cord, or thinking he's going to. Reflexes are stereotyped, meaning they occur essentially the same every time. So this reflex arc, and we kind of talked about this at the beginning, has 5 basic components. There's a sensory receptor that responds to a stimulus by producing a receptor potential or generator potential. A sensory neuron conducts the sensory impulse back from the receptor to the integrating center within the central nervous system that relays the impulses to motor neurons. The motor neuron conducts the impulses from the integrating center to the effector. And the effector can be a muscle or gland that responds to the motor nerve impulses. Reflexes can be monosynaptic, meaning reflex arcs that skip the interneuron. Remember, it can also be called an association neuron or a relay neuron. So monosynaptic arcs or reflex arcs are going to skip that interneuron. It's going to involve a receptor, a sensory neuron, a motor neuron, and the effector. Polysynaptic are going to be reflex arcs that include the interneuron in between the receptor and the sensory neuron, and the motor neuron and the effector. They can be ipsilateral, meaning the stimulus comes from one side of the body and the response occurs on the same side. The sensation's on one side, the response is on the same side of the body. Contralateral, meaning the reflex arcs are going to come from one side of the body and the response is going to occur on the other side of the body. Which do you think are faster, monosynaptic reflexes or polysynaptic reflexes? Kind of talked about this at the beginning, but what do you think? Monosynaptic or polysynaptic?
Can you repeat the question?
Of course, which reflexes are faster, monosynaptic or polysynaptic reflexes?
I want to say polysynaptic.
Why would you say that?
I think since it explains that it includes the interneuron, it'll just make it faster.
Let me say it's the opposite. Jessica, you're close, but it's the opposite, because if you don't have an interneuron, you're not going to have as much of a synaptic delay either. Does that make sense? Because
Yeah.
if there's an interneuron, there's going to be 2 synapses involved at least. If it's a monosynaptic, that means that there's going to be only one. And what it also means, let me say one more thing, monosynaptic means that the sensory neuron is going to synapse directly onto a motor neuron. So some basic reflexes, a stretch reflex. What?
I would have given her response. So you're saying monosynaptic is faster.
Yes.
I would have said polysynaptic because they're more, but that's just me.
Jessica, you're pretty smart. Because my mother just told me, she said, I remember my mom's a retired nurse practitioner. She just said that she would have given your same response. Good job. So when a muscle, what? You can be what?
Wrong together.
You can be wrong together. That's what she said. Okay. When a muscle is stretched, it fights back and contracts. I wish I had a hammer, not a heavy hammer, but a little hammer. When you stretch, this little hammer, I need to get one of these, I think. When you stretch the patellar tendon, the muscle, the sensory muscle spindles that are in the quadriceps muscles right here. These are little receptors that respond to stretch of the muscle, are stimulated via a sensory neuron that come back and relax the hamstring muscles, the semitendinosus and semimembranosus and rectus femoris muscle, and allow for stronger contraction of the quadriceps. So that's why when you hit this muscle, when you hit that tendon, it gets a tiny stretch and the muscle will contract. Mom? Quick, come here for a second, please. Well, I have a teaching assistant, guys. Give me a second. I want you, and we're gonna show this reflex because I have hyperactive reflexes, so this will be a good one. All right, guys, be patient with me here for 1 second. Hopefully my table won't collapse. We're gonna learn how to study it. Okay. She's gonna get a good hammer to bang my knee with. A sec. I need a good reflex hammer. She's not gonna bang me really hard, hopefully. So I'm gonna relax, but I have really good reflexes. I always have, so relax, relax. Did you see it? Okay, do it again. Did you see it?
Yes.
Yes.
All right, so the muscle fights back. You stretch it, the hamstrings relax underneath here. Hamstrings relax. And the muscle contracts. The quadriceps contract. Hamstrings relax. And the quadriceps contract. Helps maintain equilibrium and posture. So one is going to get excitatory. Other one is going to get inhibited. Is this a contralateral or an ipsilateral reflex? Ipsilateral meaning on the same side of the body, contralateral meaning on opposite sides.
Isperlatable.
You're right, it's ipsilateral. Because it's happening on the same side of the body. What about when you step on a nail? This reflex involves a flexor or withdrawal and a crossed extensor. So this is going to be a contralateral and an ipsilateral. It's going to be both. When you step on a nail, is going to trigger contraction of the flexors of the knee and relaxation of the extensors. So it's going to activate the flexors and relax the extensors to allow you to pull up your foot. So this is what we call reciprocal inhibition, where it prevents the muscles from working against each other. We see that here too. We relax the hamstrings and contract the quadriceps. It's a crossed extensor because what's going to occur is we're going to get relaxation of these extensors and contraction of the opposite leg extensors. Relaxation of the extensors of the foot that you've now injured, the leg that you've injured, and contraction of the other opposite leg extensors to maintain balance. So the flexor reflex uses an ipsilateral reflex, meaning relaxation of the extensors in one leg and contraction of the flexors. Whereas the extensors on the opposite leg are going to be contracted. We also call this an intersegmental reflex because the input and the output, because it also involves the abdominal muscles. Pain in the foot by contracting the foot to bring up the leg is going to contract the abdominal muscles, which is another segment of the spinal cord. So it's also called an intersegmental reflex, involving more than one segment of the spinal cord. Guys have questions for me?
I have kind of a silly question about the knee reflex.
Okay.
Is it any specific part of the knee that the reflex will show, I guess, or work? Or is there a very specific pinpointed spot?
Mom, can you answer that? Mom? Say it again, Jesse.
Sorry, is there one specific spot on the knee or under the knee to use or to hit to get that reflex, or is it any part of the?
It's one specific spot. So it's in between the tibial tuberosity and the patella.
Okay, thank you. I was just curious.
Yeah, I have what I like my reflexes because my reflexes are hyperactive. They always have been. And so I use mine because it works out so well every time. But what were you going to say, Mom?
Say, if you push on it, it will feel soft. You saw me push on it. Maybe you didn't, but you push on it to find the point, and it should feel soft below the
Patella,
below the patella, and before you come to the top of
the tibial tuberosity.
yes, that part.
Thank
Is
you.
that- You're welcome. Do you have any other questions? And you know how I said I would let you out early? Obviously, I lied. But have a wonderful few days. We will come back. We will finish covering the spinal cord, and we'll start talking about the brain.
Thank you.
You're welcome.
Have a good weekend.
You too.
Thank you.
You're welcome.
Thank you.
Welcome.